A very good morning and happy Mahashivratri to one and all. Today we'll start with the session 1 of day 3 of Gyan course, and may I now request Dr. Dinesh Handa to please start the session.
Okay, so yesterday we finished talking about metals, ceramics, polymers. They all can be used for medical implants depending on the application. I think next I want to start discussing is slowly some of the issues with medical implants. Okay, so I want to talk about proteins, and then we will talk about blood, we will talk about bacteria in the next 2-3 days. So my goal is to finish proteins today. So let's start with the proteins. Now, when we put any implant in the body, such as a catheter, the first response of the body is proteins come and adhere to the surface of the catheter. And then cells can bind, platelets can bind. If there is bacteria, bacteria can bind to the surface. That can lead to inflammation or infection or clotting or toxicity. But the first, it starts with proteins binding to the surface of the implant. So that's why proteins are very important, and there is a lot of research going on. My lab works on it also. We will discuss, I think, early next week, how to make surfaces that you can prevent proteins from adhering to the surface. Okay, it's a very challenging task because our blood has so much protein, but if we can prevent protein adhesion to the surface, maybe we can prevent clotting of the surface, we can reduce infection of the surface. So the first component is protein, right? Now, not talking about blood material interaction or protein material interaction, but what are some functions of protein in the body, right? So serve structural support role in tissue matrices like collagen, elastin, they all are proteins. Enzymes, these proteins can facilitate biochemical activities in the body, blood coagulation, DNA transcription, antibodies responsible for immune response, hormonal proteins that can coordinate messengers and signals, contractile proteins that are responsible for movement in the body, storage and transport of molecules such as hemoglobin. So proteins have so many different functions, right? So proteins are made from amino acids. They form peptide bonds, polypeptides, and these peptide bonds, just like we discussed polymerization yesterday, amino acids can be considered as monomers, and they are binding to each other and forming peptides and forming proteins. There are these 20 common amino acids, and if you see the structure here, it has an amine group, and it has a side chain, that R group, and it has a carboxylic acid group. And you might have heard some of the proteins, that amino acids that are common, like arginine, lysine, glycine, cysteine, valine, aspartic acid. They are some common proteins, common amino acids, and these amino acids can bind to each other to form different types of proteins. And I'll show you an example. Just one amino acid difference can start causing diseases and problems in the body. So there are like so many proteins, different combination of different amino acids. Everything needs to work so perfectly well, because even if one amino acid gets different amino acids bind instead of one amino acid, now you have a problem, you have a disease. And if you forgot, polymerization for amino acids, condensation polymerization takes place to form these peptides, which can lead to these they can form proteins then. Now, proteins can have different structures. there is a primary structure, secondary structure, tertiary structure, and quaternary structure. And you can imagine primary is a simple structure and quaternary is a much more complicated structure, right? So sequence of amino acids in peptide change, polymerizations of amino acids. A simple primary structure example is insulin. It has these 3 disulfide bonds, and this is like a primary simple molecule. Then the next one that's a little bit more complicated is secondary structure, and this is more alpha helix and beta sheets together, and they are forming secondary structure of proteins. The alpha helix, beta strands, and sheets turns are the most prevalent elements of protein secondary structure, which is stabilized by hydrogen bonds between atoms of the peptide backbone. And then we have tertiary structure, which is now making the molecule even more complicated. Proteins fold with polar groups on the outside, and the non-polar groups go inside when this folding happens, so that these polar groups that are outside can interact with the fluid outside, and these non-polar groups are hidden inside, which are more hydrophobic. So hydrophobic interactions, hydrogen bonds, they are common for this tertiary structure of proteins. Ionic interactions can be there also, disulfide bonds can be there. So this is just like leading to a much more, starting to make a more complicated structure in a tertiary structure of proteins. And then we have quaternary structure of proteins, and the proteins are made up of multiple peptide chains called subunits. Now multiple subunits come together to make this quaternary structure, where you have like 4 subunits coming together to make hemoglobin structure here. Okay. So to summarize, these are the 4 different primary structure, quaternary, secondary structure, tertiary structure, quaternary structure. Simple to more complicated has hydrogen bonding and ionic interactions and van der Waals. Simple ones just had those disulfide bonds. Little bit about proteins. Now proteins have conformation also, okay, and a good example of that is, see, I told you that when proteins, when there is a surface, there is an implant in the body, protein comes and stick to the surface, and on that platelets are sticking and cells are sticking and bacteria is sticking, right? But that same protein was in your blood flowing, but nothing was sticking to it at that time, right? Because the conformation of that protein was different when it was flowing, and now when it's got stuck to the surface, the conformation changed. So those receptors responsible for binding these things, they get exposed, and now all these things start sticking to that protein. So changes in temperature, pH, ionic strength, binding to surfaces, that can cause protein to get denatured, or certain property of protein that was not there, it came, or the certain property of protein that was there, it got lost. These changes interfere with intermolecular bonds that stabilizes the conformation. Proteins may not be able to revert to its original or native state after this has happened. And an example is, a simple example is like an egg, right? When you are cooking an egg, you cannot just go back now. That example I was talking about of this one amino acid difference, a common example is sickle cell anemia. You know, so glutamate is the normal hemoglobin amino acid that should be there. But if that glutamate is changed to valine, now the person has sickle cell anemia. And in sickle cell anemia, it's a genetic blood disorder where these red blood cells become rigid and they make this sickle shape. And now you can imagine these red blood cells, they are not transporting oxygen properly, and because of their change in the shape, they can get stuck in the blood vessels, which can be really dangerous. And you can also imagine, if oxygen is not getting transported properly, the patient can get a lot of fatigue and rapid heart rate, shortness of breath, because they are not getting proper oxygen transfer. Other example of proteins, one example is collagen. 25% or more of the total body protein is collagen. There are 28 different types of collagen identified, makes the structural framework for most body structures, including skin, blood vessels, skeleton. Collagen fibrils can aggregate to form collagen fibers that gives high tensile properties. Here are the 5 common types of collagen. Type 1, type 2, type 3, type 4, type 5. These are the common ones, and then there are like 20 or so more collagens that are not so important. But skin, tendon, vasculature, organs, bone, type 1, cartilage is type 2, in liver and tissue, soft tissue, type 3, epithelium, type 4, and cell surfaces, hair, placenta is type 5 collagen. Another important protein, elastin, and like the name elastin, elasticity. So tissues containing elastin behave similar to rubber band to deform and recover without damage. Okay, so it can be stretched and relaxed, and that's giving like elasticity to these tissues. Now we are coming a little bit to the point. Okay, those are good, simple little information about proteins. But now fibrinogen is an important one, because we are talking about biomaterials, implants. Fibrinogen is one of abundant proteins in blood. Okay, I think it's 3rd or 4th highest protein present in blood after albumin, IgG, and then maybe it's, I think, fibrinogen. And this is the main protein that's responsible for platelets to get activated or clot on the surface. Okay, so it's a very big protein for people in blood material interaction field. Common form is, it's a rod shaped protein, 40 to 50 nanometer in length and width of around 5 to 10 nanometer. It's 340 kilodalton. Many receptor sites to interact with platelets and bacteria, multiple cell attachment sites, and this fibrinogen protein has RGD sequence on it. Now, what is RGD sequence? RGD is arginine, glycine, and an aspartic acid. Okay, so this RGD sequence, or these 3 amino acids, form this RGD sequence. So presence of this RGD sequence on fibrinogen helps in cell binding to that RGD sequence on fibrinogen. You know, other protein, for example, albumin does not have RGD sequence, so cells usually don't bind to that protein that easily. Okay, so for our blood material interaction field little bit, albumin is maybe our friend and fibrinogen is our enemy. Similarly, fibronectin is another protein, and this also has that RGD sequence. So lot of time, if researchers are working on binding cells to surfaces, they actually coat the surface with fibronectin so they can bind the cells to the surface. It enables cells to adhere, important role in organizing the extracellular matrix. Size is around 440 kilodalton, has affinity for various molecules, contains this RGD sequence that binds to cells. protein surface interaction, adsorption of protein on the surface is the key, right? It's not absorption, it's not getting inside, it's just covering the surface of the implant. Circulation cascade, and bacteria can stick to this protein also. Like we said, that when you have any implant or a substrate in the body, proteins will come and stick to it, RGD sequence get exposed, cells or whatever, platelets, now they start getting adsorbed on the surface. And when these platelets get stuck to the surface, they start getting activated and they start calling more platelets to come and stick to the surface. And now platelets start interacting with each other, and now the clot formation starts happening on the surface. But it started with the fibrinogen adsorption that started this whole thing. So if we can stop that, we can hopefully stop a lot of things. We will talk, but trust me, people are trying everything and almost nothing works. You know, we are trying super hydrophilic materials, super hydrophobic materials, slippery materials. Proteins still eventually come and stick to the surface. Maybe for a short period of time it's okay, but for longer duration they'll just come stick. So here is an example. Somebody did the study that pre-adsorption of fibronectin that contains RGD sequence increased the adhesion of fibroblast cells, but pre-adsorption of albumin without RGD sequence almost prevented completely adsorption of fibroblast. It was only 5% cell spreading on albumin coated surface and almost 100% of the on fibronectin coated surface. What's tricky about protein surface interactions is that proteins have hydrophobic regions, polar regions, negatively charged, positively charged regions. And no matter what type of surface you are making, protein is ultimately going to come and stick to the surface. We have tried, not we, so many people before us have tried so many things. We have tried things. Protein ultimately gets stuck to the surface. Now, these three scientists have been pioneers in modern protein adsorption studies. So Leo Roman, he's no more, but he has done a lot of work in protein surface interactions, and we will talk. There is a Roman effect also. We will talk about it. That's he figured that out. Dr. Tom Horbett, he's emeritus. He was a professor and now emeritus at University of Washington. Lot of work in 1990s around that time. Another person that I didn't put the picture here is Dr. Buddy Ratner. I don't know if you have heard his name, probably because he has done a lot of work on blood material, protein material interactions, understanding. That big book that I showed you in the start that you can buy, that's the latest version of the book. But the earlier version of the book, Dr. Buddy Ratner was the one who compiled the chapters in that book. Now other scientists are making it more broader, but he was the original person who started it. Dr. John Brash, he's a professor at McMaster University, and I had the privilege to meet with him maybe 6-7 years ago. We had a big small conference discussing blood material interactions, and I met him there. He has done a lot of work on protein material interactions also. So I'm telling you these names so that if you are interested in the field, if you search their names, you might get some really good basic papers that they were working on in this area. When you have protein surface interactions, a lot of things can happen, okay? Proteins can adsorb on the surface, they can desorb from the surface, but I'll tell you one thing, desorption of protein is not very common. Once the protein gets stuck to the surface, it's not very common for the protein to detach from the surface that easily. Because there are so many interactions happening, hydrogen bonding, hydrophobic interactions, that protein gets stuck so tightly to the surface that it's not very easy to physiologically just remove it from the surface. Fractionate mixture of proteins can get stuck to the surface. Proteins can displace some other proteins, not very common, but it's possible. We will talk about it a little later. And actually this phenomenon is known as Roman effect. They can reconfirm, the conformation of the protein can change. They can orient in certain way. They can get activated or they can get deactivated after they get stuck to the surface. Another thing, protein adsorption is rapid. So as soon as you put implant in the bloodstream, let's say, protein adsorption will happen very fast. Okay. And once it gets stuck on the surface, it's not easy to remove it also. So if you are designing coatings, your coating better starts acting very fast. Otherwise, it will just take over. Okay. I can discuss this later, but let me mention this now. Another issue is sometimes, anti-fouling surfaces are great, but let's say you have a surface and it's preventing protein adsorption, it's preventing clot formation to some extent, it's anti-fouling, but it's not completely anti-fouling. That can be a problem also, because if something got stuck to the surface but it's very loosely bound, because your material is preventing that binding to happen, now that can cause embolism. And what is embolism? So if something gets stuck to the surface, let's say platelets, proteins, there is a little clot, it comes off because it was not that tightly bound, and now it goes in the bloodstream. Now that can be even more dangerous. So making anti-fouling surfaces is great, but if your surface is somewhat working but not properly working and it's not completely anti-fouling, things are still binding, but they got bound and clumped and then got detached, now that's not very desirable. Another thing is protein adsorption on surface, it plateaus after certain time, right? It's not that you're going to keep getting protein stuck, right? With time, protein adsorption will happen, but then it will plateau. Similarly, even if the concentration of your protein solution is very high, it's still going to plateau after certain time. Now, most materials absorb same amount of protein, or they form monolayer. Okay. Now, depending on the surface, different types of proteins might come and stick to the surface, right? But it's still going to be a monolayer, and approximately that 100 to 200 nanograms per centimeter square protein will get stuck to the surface. And you can imagine now, I hope, even with this much that we are talking, that how challenging all this is, right? How to prevent, blood has so much protein, the protein is flowing on your surface, you have an anti-fouling surface, but it needs to keep continuously fighting that protein in the bloodstream. This just shows that amount of protein of surface isn't proportional to the concentration of the solution. It's just going to plateau at some amount. and protein continues to adsorb until at least a monolayer is formed. Oh, so this work is Dr. Horbett's work that I was showing you in 1996. And see, we also need to always appreciate that is around 30 years ago, and maybe the facilities and understanding of the field was so much less than what it is now. So things done at that time, it's even more, we have to appreciate all the work that was done at that time. I was talking with some students today earlier. Sometimes we feel the work I am doing, it's not so impactful because it's not bringing something to market. But we cannot forget that work we are doing, maybe it's just leading to a publication, but 5 years later somebody can pick up that work and take it to the next level, or 10 years from now actually that can become a product because we helped them understand something. So even if we are designing something today, maybe it's Dr. Horbett or Dr. Roman or Dr. Brash's work or Dr. Buddy Ratner's work that's helping us make the path to come to market with these devices. This is just showing using AFM fibrin adsorption on the surface. Have any of you worked with AFM? So a lot of my PhD was using AFM or atomic force microscopy. It's a pretty cool instrument for multiple reasons. One, so AFM has a small tip and you have a surface and you are scanning the surface. So you can actually with the AFM tip look at nanometer structures on the surface. Okay, so that's cool. But even better is you can do AFM imaging in physiological conditions. See, lot of proteins and all these physiological components, if you dry them, they get denatured. And now if you're imaging them, you're not getting the true picture or true information about it. But with AFM, you can use the buffer and the temperature, physiological conditions, while you are even imaging those structures on the surface. So it's a very, very powerful technique. Another benefit of AFM is, and it actually relates well with the protein, you can measure piconewton level forces with AFM. I have not done AFM in last 12, 13 years. Just last year we bought an AFM for our lab. I am talking with what I was doing 12, 14 years ago. I'm sure AFM has so much more capability now. So what do you mean by we can measure piconewton level forces? So for example, I don't know if I have A, B, C, D, 4 surfaces, which one binds fibrinogen strongly? I don't know. So maybe we can put this fibrinogen on the tip of the AFM, and then you have these 4 surfaces. You take the tip down, press it on one surface, and then try to pull it. Machine is doing, AFM is doing all this, and you can measure the forces needed to pull it back, detach it. You can do it with all the surfaces, and now you can see for which surface you had to apply the highest force to pull it. So now you know your surface A attaches fibrinogen more strongly than B or C or D. And you can do all this in physiological conditions. So really cool way to understand protein surface interactions. Then I said it a little bit. This study basically showing a lot of people have done it. This is irreversible. You adhere a protein on the surface, then you try to wash it off. And I think Dr. Buddy Ratner, Dr. Horbett, their group did this work also. You cannot wash it off now. it's stuck to the surface. And when in my own way to understand this is, think of like a Velcro maybe, a protein, let's say fibrinogen, it might have these 500, 700 Velcro type interaction points, and now your surface, it got stuck like that. Now you can flow a liquid on it at a very high speed, but there are so many spots on that protein that it's stuck with the surface. Even if you can get detached 2-3 spots, it's still stuck 100 other spots on the surface. So now the protein is stuck on the surface. It's very difficult to just wash it off. Another thing that happens is proteins compete with each other to get stuck to the surface. Depending on the surface, which protein will come first or faster, maybe more abundant protein can come first, maybe a protein that's smaller in size can come first. If a surface is more hydrophobic versus hydrophilic, which protein is coming and getting stuck to the surface might change. But like we said earlier, amount of protein getting stuck to the surface is approximately in the same range. Okay, Roman effect. So basically the Roman effect is, in our blood, albumin is the highest amount of protein is albumin. Okay, fibrinogen is number 3, I think, or number 4. And I think it's 10 times less than albumin, fibrinogen. Albumin is much smaller protein compared to fibrinogen. And albumin has lesser contact points, maybe 10 times less contact points compared to fibrinogen. Okay. So what Roman effect does is, as soon as a surface is implanted, albumin is rushing to the surface and getting stuck to the surface because it's smaller and it's more abundant. Later on, fibrinogen, which is a much more bulkier protein and it has more spots to attach to the surface, it can come and remove albumin and it takes its place. So this is Roman effect. So highest mobility protein generally arrives first, later replaced by less motile proteins that have higher affinity for the surface. And typical example is fibrinogen coming and removing albumin. And this is our problem because fibrinogen is not our friend when we are talking about clot formation or blood getting activated on the surface. Okay, so some facts about proteins. Adsorption is rapid. Within seconds, proteins can come and adhere to the surface. Proteins compete with each other. Plateau level adsorption happens. Mostly monolayer adsorption happens. Mostly the adsorption is irreversible. Yes, Roman effect can happen, protein displacement can sometimes happen, and adsorption can cause denaturation of proteins. Now we have this really cool paper we can discuss. Let me tell you a little bit about Dr. Robert Latour. He is a good friend and a mentor. He just retired, I think 3 years ago, from Clemson University. He has also done a lot of good work with protein material, blood material interaction type work. And Dr. Shivaraman, he is a friend. When he published the paper, I didn't even know him in 2010. I read the paper, I loved it. 2-3 years later, after this paper was published, I met Dr. Latour and Balakrishnan in conference and we became friends, and Dr. Latour helped me a lot for last 10 years before he retired. I would like to discuss this paper because you guys will like it a lot. But before that, I wanted to also ask, do you guys know, I hope you understand impact factor of journals, right? Anybody doesn't? Okay, great. Do you guys understand H index? I ask this because, the class I teach, a lot of students don't understand H index. Some people like H index, some people don't believe in H index. I don't know. It's not perfect, but it's something, some way to. Now, one thing is number of citations, right? Somebody got cited 2,000 times or 5,000 times or 50,000 times. So that's fine. But the problem there is, okay, what's the problem there? Can somebody tell me? Why can't we just say number of citations? Why do we need H index? If we just look at total citations, right? Let's say I published one amazing paper that everybody is citing left and right, but all my other papers are not good at all, right? Now I have 5,000 citations, but 4,500 are coming from one paper, and all my other work is giving me 500 citations, right? So that's why H index can be useful. Now what H index is, it's a little bit difficult. Without a board, it's even more difficult, but please listen and hopefully you will understand. So H index is, let's say somebody has published 5 papers, okay? And their one paper is cited 100 times. Their second paper is cited 1 time. Maybe you guys can write it so that when I'm telling you, you will understand, because I might forget while saying all this also. Okay, so 5 papers total this person has published. One paper published 100 times, second paper published 1 time, third paper published 1 time, fourth and fifth cited 0 times. So 100 citations, 1 citation, 0, 0. What is this person's H index? So this person's H index is 1. Okay, and I'll tell you why. Now let's say this person's second paper, one is cited 100 times, second one is cited 2 times, third paper is cited 1 time, fourth is 0, fifth is 0. Now this person's H index is 2.
The minimum number of times.
Yes. Now for this person to become get the H index of 3, their 3 papers, each one should be cited at least 3 times. And to get a H index of 20, you need to have at least 20 papers, each cited at least 20 times. Now if one is cited 1,000 times, it doesn't matter. 20 cited 20 times, then only your H index will become 20. Okay. So, I mean, I thought you guys should know, you all are in science field, right? So, it's not everything, but it's some way to see that, right? Now, there is another one that's called I-10 index. Okay. So, I think that's also good because that's saying how many of your papers are cited at least 10 times? How much of your work is cited at least 10 times? It's I-10 index. So I feel the benefit of maybe I-10 index is it's showing little bit future trajectory of the person maybe, right? Because if you have 10, at least 10 times cited lot of papers, maybe it's showing in coming years, they will just keep going up. And it's also a little bit showing overall impact of your work that, okay, getting a lot of citations is always great, right? But then it's also showing if you're publishing a lot, are others citing your work? But I have also heard from senior people, sometimes it's not, because what if you are a pioneer in a field? Nobody is citing you because nobody is at the level that you are at. So it's sometimes not true also, this H index, right? Because you are running ahead of everybody else. It might take few years for people to start citing you when they start working on that field. Or your field is so difficult that very few people are working in your field. Now you're not getting a lot of citations. So it's a good number to look at, but you cannot just totally rely on that number. Okay, so this paper was published in Biomaterials, which is a really reputed journal in our field. Maybe right now its H index is 12 or 14 or something. Oh sorry, its impact factor is 12 or 14 or something. So really reputed paper in our field. So what they did in this paper was, maybe read this abstract little bit. So when we talk, what they did is they used different surfaces with hydroxyl group, amine, methyl group, and they put different amounts of fibrinogen on it. 0.1 milligram per ml solution, 1 milligram per ml solution, 10 milligram per ml per solution, and you can see that how the amount of protein absorbed was so much higher with 10 compared to 0.1, right? Makes sense. Then what they did was, using circular dichroism, you can see how much alpha helix and beta sheets are there in that fibrinogen. Okay. So the first A is 0.1 milligram per ml fibrinogen solution, B is 1 milligram per ml solution, and C is 10 milligram per ml solution. Okay. If you see here, on different surfaces, native fibrinogen, without putting it on the surface, had lot of alpha helix. Okay. Once you put it on the surface, hydroxyl remains almost similar, but other surfaces, amount of beta sheets increase after you put it on the surface. Okay, we are looking at A, which is the least concentration of fibrinogen solution. Same thing happened for B. When you put 1 milligram per ml solution, again alpha helix little bit reduced and beta sheets increased for the same fibrinogen when you put it on the surface. Okay. But when you see number C, the C part, it didn't change. All the alpha helix remained very high and beta sheets remained very low, no matter what surface they tried. Why? Saturation, right? So can you explain a little bit more? You're almost right.
So it's independent of concentration but depends upon the conformation?
Yes. So when they put so much fibrinogen on the surface, there was no place for fibrinogen to open up. So it stayed in its almost native state, right? But then the other interesting thing happened. Now what they did, they put same amount of platelets on all those. So they took the surfaces, they put fibrinogen very low to very high, and then they put same amount of platelets on all those surfaces. And very counterintuitive, the highest fibrinogen surface had least number of platelets. Okay, and the reason is this: because in native fibrinogen state, it's not binding to platelets. The conformation is in such way, alpha helix is high, beta sheet is low. And after we put that high concentration on the surface, we stayed in that ratio, and now the RGD sequence or receptors that were recognizing the platelets, they were hidden and platelets couldn't stick to the surface. So very basic such study, but very well done and very informative, proving the point. And we have already talked about this again. I'm mentioning that this is the issue. So protein adsorption, and it's an entropy driving force, right? Proteins liberate structure, water, and that's why they want to just get stuck to the surface. And that's why a non-fouling surface lot of times are very, very hydrophilic, because they bind water so tightly that protein cannot come and just remove that water and takes place, right? The only thing is, for how long? For how long we can just keep fighting that tightly bound water on the surface. Okay, you guys can help me answer this. The protein is so hydrophobic. In a situation, water is bound so tightly that protein is not able to stick to it. But the hydrophobic surface, due to hydrophobic interactions, it's easy for hydrophobic protein to get stuck because water is not bound so tightly on the surface. But you are right, that is more hydrophobic, that is more hydrophilic. Now, when you have a surface can have hydrophobic regions, polar regions, negatively charged, positively charged. So depending on how the surface is, the protein can adsorb in different orientation, right? Okay, so question for you now. Take 5 minutes, write it down in your notebooks. What are some properties of proteins that affect their interactions with surfaces? What are some properties of proteins that affect their interactions with surfaces? Okay, can somebody tell me one property at a time?
Structure of protein.
Structure of protein. Okay. What else?
So you also mentioned the binding site should be open. The nature of the protein. Also. Amount of protein that is being binding.
It's like properties of protein, so not just amount.
Orientation.
The region specific charge.
Charge of protein, yes. What else?
Steric hindrance.
Steric hindrance. What?
Isoelectric point without pH or isoelectric point.
That's under charge. So charge, yeah.
Contact angle.
Contact angle. It's how will you measure contact angle of, so something else.
It gives free energy. Hydrophobicity.
Hydrophobicity of protein, charge of protein. Yes, there are some things we already talked today. Yes, but there are things we talked today. There are simple things. See,
Interaction. Hydrogen bonds or disulfide bonds.
okay, so if there are hydrogen bonds or disulfide bonds, how will it impact protein's interaction with the surface?
Make more surface area.
or can we say in easier way, unfolding rate of protein? Because if a protein comes on the surface, it's sticking, the faster it unfolds, faster it grabs onto surface, and now the other protein cannot come and remove it. But it takes more time to open, something else can come and kick it off, right? Okay, there is one more that you guys are missing, and we talked about it today.
Cell binding.
Size of the protein, right? If the protein is very big, it's going to take time for it to come. It might not come, reach, something else has grabbed that spot. So, size of the protein, and bigger proteins have more binding sites also, right? It can come, so a smaller protein can come faster but binds less tightly. A bigger protein can come later but can bind more firmly to the surface. Charge, isoelectric point, like you mentioned, it can absorb more to the surface. Hydrophobicity, more readily absorb on hydrophobic surface. Structural stability, it can fold to a greater extent and form more contact points. And then of course unfolding rate, faster it unfolds, faster it can get firmly stuck to the surface, right? Now, here is another interesting thing. Albumin is almost 4 to 5 times smaller than fibrinogen, 67 kilodalton versus 340 kilodalton. Albumin has 10 times less contact per molecule compared to fibrinogen, right? And all this impacts lot of things like we talked in Roman effect. But albumin is bigger or fibrinogen is bigger? Fibrinogen is bigger. Albumin is more abundant or fibrinogen is more? So that's more abundant, it's smaller, but it does not bind so strongly, so fibrinogen can come and easily replace it. Okay. We little bit talked about it, but this is again showing that unfolded protein lightly expose more sites. Factors that affect rate and extent of unfolding can result in greater surface activity. Less stable proteins are those with less intramolecular cross-linking are likely to unfold more and faster. Okay, protein denaturation on surfaces. Protein structures are not rigid. Proteins are flexible chains that have been coiled, folded and bent to assume particular 3D structure. Changes in microenvironment of protein can alter the conformation of the molecule. So in solution, we talked about alpha helix and beta sheets little bit, versus adsorbed proteins can be very different. Alpha helix going down, beta sheets going up. Upon adsorption to a surface, conformation changes, but the protein usually retains some of its biological activity, not all of it, but maybe to some extent, but some is lost. This is just showing that how, if we see the last one, how based on the surface, the difference in protein conformation. You know, for some protein is binding from some way, in the other one, if a surface is more polar or non-polar or negatively charged or positively charged, same surface protein is binding, but the conformation and the way it's binding can change. So Professor Sidlicki is also my very good friend and a collaborator. We have published together. Maybe I'll discuss it next week about some of the work we did together. but this work he did a long time ago, I think. Basically, it's showing, again, it's AFM, atomic force microscopy. Actually, can you tell me what it's trying to show on that side? The right side? mica, APTS, OTS.
So the surface structure of those wavelength.
So that thing, that bright thing, that's fibrinogen. Okay, I can tell you that.
The topography of the
Sure, yes. So AFM tip is imaging the surface, and that's fibrinogen bound to the surface of mica, APTS and OTS. But what else can you tell me from that image?
So in which there is the height of the topology is more, where the fibrinogen is not binding more closely to the surface, whereas in the third structure it's diffused, where the fibrinogen is binding the highest among the given surface.
So the third one you are saying it's binding more.
Because the tip of the AFM is catching it to be diffused, not much pointed as in mica or EPTS.
Yes. So mica is a very hydrophilic surface. You know, if you put a drop of water on mica, it will just, so the surface is very hydrophilic. OTS, silane, it's a very hydrophobic surface. So it's basically showing that when they put fibrinogen on this OTS surface, fibrinogen due to hydrophobic interactions, and mica had tightly bound water, it's difficult for fibrinogen to stick to it so nicely, but on OTS this fibrinogen can stick and nicely expand. And if you see, the length of fibrinogen here is longer compared to these two. So it's like more opening up and binding it with more contact points. How many contact points fibrinogen has?
Just 770 something.
700 something, right? So more of those contact points are grabbing onto surface now because of surface hydrophilicity.
It's also unfolding more in OTS.
It's unfolding more, that's why it's spreading more, right? So now if I put platelets on these surfaces, which one will have more don't answer, I'll ask somebody to answer. Okay, so if I put same amount of platelets on all three surfaces, which one will bind more platelets now? Who wants to answer? Raise your hand. Okay, so I'll ask somebody who didn't raise the hand. Okay, what do you think? Which one will have more platelets if I expose same amount of platelets to all three surfaces?
if you expose more amount of platelets, I think the OTS will find it more.
Why?
Because it is more hydrophobic.
So let's say I put lot of fibrinogen now. So forget OTS's hydrophobicity. I covered OTS with fibrinogen. Like now we cannot see OTS anymore. It's covering with the fibrinogen for all of them. Now which one will bind most platelets?
So in that case, if OTS is covering, if fibrinogen is covering all of the surface, so then the binding of platelets on OTS will be less, because the original availability would decrease. I think what I understood.
Okay, anybody else want to?
So isn't it about the surface, like it's spreaded in the third of
Okay, so if I put same amount of platelets on all,
more surface to cover, so platelet will bind more in like,
to which one?
OTS
OTS, because
surfaces.
since fibrinogen open up more, conformation more, those RGD sequence are exposed more, now platelets can go and grab onto them. But the other one, since it was hydrophilic, fibrinogen is not liking the surface, it's little bit still closed, and now there is lot of fibrinogen, but they are not exposing their receptors. So platelets, it's not easy for platelets to get stuck to the surface. So OTS will bind most platelets out of the three.
Is it also without fibrinogen, like in the blood clotting mechanism, the platelet won't bind? So that way also, in which material the fibrinogen is binding the most, the platelet will bind there also.
So, and if you want, we can discuss later if you want to discuss after we are done. Okay. Now, next little assignment, you can write the question. Properties of surfaces. Earlier I asked what are the properties of the proteins. Now, what are the properties of surfaces that affect their interactions with the proteins?
Also, what's OTS?
It's a silane. Both are silanes.
Kind of
They both are silanes, yeah. So you coated the surface with one type of silane, another type of silane, and maybe they have different contact angles, and I feel OTS contact angle is more than APTS. It's more hydrophobic than APTS.
So in this case, like while they are not growing, so in this case we will choose mica for as our surface because it has less fibrinogen and less platelet activation.
Okay, good question. So I think when they published this paper, they were not thinking about medical devices or anything. You know, they were just trying to do a more mechanistic study to explain this point. You know, see, the understanding we have now, such strong understanding, it's because of studies like that. So we are not making a medical device of mica. Okay, we are not using mica in making medical devices. Yes, but yes, if you want to make a surface that's more hemocompatible or prevents clotting, we are choosing more hydrophilic surface because this one will chances of clotting is less. But then next week we will talk a little bit about super hydrophobic surfaces. Okay, now if the contact angle of the surface is greater than 150 degrees, it's called super hydrophobic surface. Now the point is the surface is so hydrophobic, like the leaf of a plant or something, that if you drop blood, it just rolls off. So now you are preventing clotting by totally opposite way, that things are not sticking to it because of air pockets and air trapped there, that you are just rolling things off. So it's not that, oh, hydrophobic is bad. Maybe super hydrophobic is good in preventing infection from bacteria or clotting.
So when we say protein interaction, we are aiming the protein should not stick there.
No. So I'm saying properties of surface that affect their interaction. How can I say? I'm not saying protein should or should not stick. So you are saying, oh, I don't want to tell you the answer. If the surface has more of this, it will affect the interaction. It will interact less or more, it's okay, but this will impact the interaction. So see, I love this field because the problem is so simple, but it's not simple at all, right? How to make a non-fouling surface, how to make an antibacterial surface. It sounds simple, but people have tried everything. You can go on Google Scholar, almost every surface has been tested. Okay, who can say the first surface property? Okay, so hydrophobic and smoothness. So topography of a surface, right? So what will smoothness or roughness will do? If you have a smooth surface versus rough surface?
No, the protein will not adhere
interact with it more, and the rough surface will interact more.
So rough surface, more protein will get stuck, and smooth surface, less protein will get stuck. And why is that?
Due to the contact that will form between the protein and the
Or the surface area is increasing, right? So that's why you'll bind more. Okay, what else?
So lattice shape for the crystalline structure of the metal.
Sure. So I think you guys almost answered. So topography, composition of the surface, chemical makeup of the surface, hydrophobicity and heterogeneity of the surface. See, a surface is not necessarily looking like all same. It can have different domains on the surface. Some domain might interact more or less with proteins. Some domain might want to absorb fibrinogen more. Some domain want to maybe absorb albumin more, right? So different domains can interact differently with the surface also. Okay, surface topography, charge, chemical composition, geometry. Surfaces with more topography feature will expose most surface. Surfaces with grooves or pores have higher surface area compared to a smooth surface. Other surfaces causing roughness. So how you are making a surface, I'll show a slide in five minutes. That changes how the surface looks also. You know how you're machining the surface. So that can change the topography of your surface. Chemical composition, if there is an oxide layer on the surface, that can sometimes passivate the surface. Ceramic and glasses contains metal and non-metal ions, which can impact how it's interacting with the surface. We saw polymeric surfaces in Dr. Latour's paper, hydroxyl surface versus amine surface. They change how protein adhere to the surface. So hydrophobicity, hydrophilicity, hydrophobic surface typically absorb a monolayer of protein. But one question is, can protein monolayer passivate the surface?
So that is oxidize the surface.
No, not oxidizing, but can you have a surface that absorbs the protein in certain way, in certain conformation, that now things are not sticking to it?
Okay, like evenly adhered.
Yes. You know, protein conformation changes can occur. Super hydrophobic are anti-fouling and repel everything. You know, hydrophilic surface hold water tightly. Proteins can't remove this tightly bound water. Bound water acts as a shield or barrier. Proteins in native conformation. Now protein is not like getting converted to that alpha and beta sheets. It's staying, it's not happily spreading on the surface and sticking. So more beta sheets are coming in terms of that fibrinogen example that we saw. On microscopic scale, biomaterials are inhomogeneous. There can be patches, polymers have microstructures, and all this can impact surface. You know, so many times when we make coatings, films, you look under SCM, surface looks great. You look under SCM and there are certain things, features on the surface. This is just example. Don't worry about all this. Basically what we are saying is it's a titanium alloy, but how it was formed, the surface looks very different. This is an example that you have this polyethylene surface, but lamella splitting, or you know, yesterday we talked about how there is a crystalline portion of the polymer and amorphous portion of the polymer. That crystalline portion has these polymer chains stack with each other. If that breaks or splits, it's causing features on the surface. And now that feature can start more proteins to get absorbed, and that gives place for platelets to get activated, and then it can start recruiting more platelets, which we'll talk tomorrow how clotting happens. Here is an example of a styrene butadiene block polymer. It has different domains on the surface now. And now each domain is interacting with the surface in a different way. Now, factors that influence protein surface interactions, protein properties, we talked size, charge, structural stability, unfolding rate, surface properties, topography, chemical composition, hydrophilic or hydrophobic regions, heterogeneity of the proteins. Okay, so I want you guys to think about it. When we are talking about protein surface studies, let's say your research is my area and you are designing surfaces, you want to understand protein surface interactions. What are some of the studies that come to your mind you would like to do? Think, just jot down some points, think for 2 minutes, and then we can discuss.
Studies as an experimental study.
Experimental studies. I know some of you are computational, but I'm just thinking more bench. On bench, what type of studies would you want to understand your surface? What type of studies would you want to do? Reactivity of protein. Okay, what do you mean by reactivity of protein?
Functional group present in the protein.
Sure. So what would we do to study some of those things?
We will read about the structure of protein, the functional groups, and their reactivity.
Okay. And what will we do bench on the bench? What would we do? How would we do some of those studies?
By structure, oxidation of protein.
I already discussed a lot of this today actually. You guys just have to think what we talked and you can answer a lot of these questions. See, I know you guys are simulation. I'm saying what will we do bench top study?
So extra diffraction, AFM.
Yes. And with AFM, what are we studying? What would you study?
So the surface properties from AFM and maybe the protein properties from the X-ray diffraction or the NMR studies.
NMR mass spectrometry.
Okay. Anybody else?
So we can use TEM to study surface.
Okay. And if we forget the instruments, what would you want to study? Like, I say, okay, my lab is designing this new surface. And I'm saying, oh, my material is anti-fouling or not. What all studies would you want to do on my surface? Forget what instruments you will use. You want to do adhesion. You want to see what protein sticks more and what doesn't stick. Okay. What else?
So there was this diagram graph which AFM, when you talk about if we have a layer of fibrinogen on a surface, so through that like we can go for albumin and we can check for different proteins, like how does it
So
I don't know how to like name it, what
really good. You want to do competition study, right?
comparative.
So you are competing, you are showing, can you put albumin and fibrinogen together on the surface, and then you want to see on A surface what is sticking more versus B surface. What if you can design a surface that sticks more albumin than fibrinogen?
Take into account the orientation, the angle.
And then if it's sticking, what's the orientation? So yes, so we want to study orientation, we want to study competition. What else? We want to study how sticky different proteins are on different surfaces. Bonds, okay.
So can we go for like antibacterial here?
We will go tomorrow. No, Friday antibacterial. Today let's be anti-fouling.
So I would like to study like the blood flows drains the entire body. So I would like to go into the physiology and check why doesn't it coagulates anywhere else in the body. Like it flows through the bone marrow, through the tissues, through the ligaments. So there must be a common property lining all such physiological structures and then try to
So you're
replicate
saying
it
why
at some
blood
methods.
why blood is not clotting at other places?
Yes, sir. Like not adhering in
That's
other
our
places.
Monday's talk.
Yeah.
So that's what my research area is, to study what body is doing
Yes, sir.
and why can't we make surfaces like that so we do the same thing what body is doing.
Exactly.
Yes.
I think this is a straight way, a very laborious way, but the body will only give the answer. Develop such a service.
It has given the answer. We are still trying to make it work, but we know the answer.
So
Okay.
I would like to study that.
Okay.
What's inside body.
What else?
So can we go for a combination of surfaces? Like two materials?
Can you elaborate?
Like in that figure, mica, interfacial or
Oh, you have domains, different domains.
so different proteins will react differently, so with different orientation and with different degree of binding.
So, thermodynamics.
Can we go for like that?
So, my worry there is, and maybe we can, what I'm just thinking quickly. So let's say you have one domain here, right? One protein is sticking here, one protein is sticking here.
Like when we have taken OTS.
Yes. How is it helping? Think of a medical device, right? You have certain portion of medical device, one domain, certain portion of medical device, one domain. One is sticking more protein, one is sticking less protein, or one is sticking one type of protein, one is sticking one type of protein. How will it help? Let's say you say, yeah, one domain will stick less protein. Then why shouldn't I make my whole device that domain? Why should I have another domain there? And maybe you have some thought there. This is just immediately that comes to my mind, right? Okay, let's come here. So amount of adsorbed protein I want to understand. Am I making surface that has less surface? Desorption and displacement of protein. Am I making surface that proteins can easily detach? What is the orientation? You guys said protein activity. What if I have designed a surface that after protein sticks to it, the conformation is such that platelets are not sticking to it anymore? That will be cool, right? Thickness of the protein, competition. Can I displace one protein? Can I put albumin more strongly than fibrinogen? Then my problem is also solved, right? Okay. So this is our last slide for today. Just to give you an idea, there are a lot of different techniques used to study protein surface interactions. You know, you can, let's say you have fibrinogen stuck to the surface, you can get an antibody that binds to fibrinogen and it has a fluorescently labeled that you can check. You can have fluorescently labeled fibrinogen that you can buy, and then you can put it on different surfaces and do fluorescent imaging to see what surface is more. You can do QCM or quartz crystal microbalance where you can check the weight. You can do AFM. So there are a lot of different techniques available to just get in-depth understanding of these protein surface interactions. Okay. So I'll stop here, but we can still discuss if you have any questions. But just to summarize everything, for a medical implant, protein is the first thing that comes on the surface. Tomorrow we will talk about blood and platelets, how they come and stick to the surface, but protein is the first thing. Making anti-fouling surfaces is very desirable. Can we prevent protein adsorption from happening? Certain proteins bind more strongly. Roman effect can happen. Fibrinogen is our enemy because it binds to platelets more. Albumin is smaller, more abundant, but it can be replaced by fibrinogen. Certain properties of proteins and surfaces impact their interactions with each other. I think more or less that's what we discussed. Okay. Questions? But we will do antibacterial, we will do learning from the body, all that is coming.
So, when I can remember that I was reading this paper that was published in Science back in November last year. It was exploring the nature of piezoreceptors on this on our gut, small intestine. It said that where there is more mechanistic stimuli, and where in the regions where the piezoreceptors are more frequently stimulated, that those cells have better chances of regeneration and proliferation and everything. So if that is the case with the specific to a receptor, that might be available to the entire all in each and every cell. I might not be sure about this. I feel might be wrong. So is there something that we can do with our implants, that they give some electrical or certain mechanical stimuli to our specific region that stimulates the surrounding tissue to bind more efficiently to that?
So I know for hip implant type work, you can maybe try these type of things. But let's say you're putting a catheter in somebody. You don't want fouling to happen. So how do we prevent fouling? To increase cell interaction, we can make surfaces porous, like hip implant type work. But the challenge is, how can we put an implant in that does not interact? Like the body, the blood can keep flowing without basically interfering with that tube. That's the challenge. So like she said, blood is flowing in our blood vessels so nicely. We are not getting any clotting, otherwise we'll die. It's flowing through tiny blood vessels, no clot happening. But as soon as we put any tubing, a catheter or a stent or a vascular graft, blood wants to attack it. Proteins want to attack it. They want to stick to the surface. How can we make a surface that stays free of all that?
So but
That's
that's more of a logical response. Like we have a
Yeah.
cut or something.
Yeah.
So it's human understanding that we are inserting a catheter or something, but the body
Yeah.
is taking it more like a foreign invasion. So it does activates all of the immunity, neutrophil, transmigration, everything. So we need to mimic something which the body considers it's very low.
Yeah.
For that particular. So what if we start making catheters, which itself is a biomaterial, catheter made of a tissue,
Okay.
which the body recognizes as its own, and maybe we can take, for example,
So one,
a cell of body.
how will you get certain strength in the material? How will you sterilize that material? How can you store that material? Because see, in hospitals, what doctors want is, here is like surgery going on, here is a shelf full of catheters, they'll quickly grab it and then they, even putting a catheter in a freezer and asking a nurse go get a catheter in that time of surgery is,
Risky.
it's too much time and chaotic. They just want very easy process when they are doing it. I mean, this is how we need to think. Yes, you are right, but it's still not so practical solution. But this is the next 2, 3, 4 classes are going to be about. So think about it more. Maybe you guys will give me some good ideas that I can try. So yeah.
So like the umbilical cord, so it stays active even after some time. Like I know people who delay the cord cutting for the genuine transfer of all the immunity, and all, even after the birth, given that the baby and the mother are comfortable and everything.
Yeah.
So the material of that umbilical cord is made,
Yeah.
and it stays good even in that environment. I know we don't have to insert it back or something, and the baby don't have to carry it whole.
But think something like that when you're designing the material.
Yes, sir, similar, which is not much prone.
Yeah.
Obviously, we can't keep it here on the table and then take it for operation. It must be immersed in some fluid for that particular thing, and just the way taking all the precaution we saw yesterday in the hip replacement,
Yeah.
it must be inserted.
Yeah.
So for that example, elastin in our aorta and that cardiac wall,
Yeah.
everything. So it's non-leaky, it's very mechanical, it can take a lot of pressure inside. So what if we make a catheter or something of elastin or maybe a biomimic of it, and those tissues can be easily grown in lab.
Yeah.
now from the
But see,
scientific
again, you have to think, can we get the mechanical strength?
Yes, sir.
Can we sterilize it? Can we store it for 2 years? Does it have storage stability of 2 years? Because see, I don't know what's the exact number, but let's say 1 billion catheters are used every year. And nobody's saying, I need catheter tomorrow, let's order catheter, this special catheter to come next week, right? I'm going to buy catheter for next 6 months or whatever time supply and put it on the shelf.
So it would take a lot of time.
And how much will it cost? See, your idea might be really cool, but if the cost of the catheter is 100 times more, okay.
This is like a scaling problem. It's a business problem.
It's
a science
everything,
problem.
you know, it's a science problem, but it's a business problem.
Yeah.
It's everything. And you are thinking more practically, right? That's how startups, commercialization, you can still publish a cool paper by saying, hey, here is my elastin material, here is my cool catheter. You might get a really good impact factor publication, but that's it. There is no future after
There's
that.
no bench to bedside.
Yes, yes. Okay, guys, thank you so much. And yeah.